Yeah, so we're looking at recirculation with hybrid treatment systems, and specifically looking at the removal of PGRs and pesticides by those systems. I'm trying, okay, all right, it's working. So the project objectives are to use the existing hybrid treatment systems to look at the effect of the media that we're using in those systems to remove plant growth regulators and pesticides, to look at the treatment sequences, how that affects the results, and a number of operational parameters, in particular temperature and hydraulic retention time. And of course, there's a bunch of knowledge transfer going on. And the other thing that we've tacked on to this a bit is some continued monitoring of the existing permanent treatment systems, because we'd like some long-term data. So we've tried to fit that into the project as well. So why are we doing this? So I like this slide because it's a survey that was done in the States of growers, and the question that was asked was, what are your concerns about recirculating irrigation water? And the top three were pesticides, including herbicides, plant growth regulators, plant pathogens, and nitrates and phosphates and salts. So that's what growers were worried about, and that's what we wanted to look at. So we had a first project where we had these portable pilot units built, and there was also 2 permanent systems involved as part of that project. And so this is what the portable units look like. They're quite large. They're in container shipping containers. And the inside is a series of cells with various media in, and then our permanent systems are a lot larger. And there's 2 of those that were built as part of the system. And in that first study, what we showed was that these systems are capable of removing nutrients and plant pathogens, specifically fungal plant pathogens. That's what we were looking at. So that was the first study that we did, and this is this COHA-ACHO is continuation of that. So as I said, we're in the 3rd year of this project, and so our focus this year was to look at the series studies to test the media sequences and the operational parameters. We use lab analysis at the University of Guelph. And we're also doing bioassays. There's steering committee meetings and KTT events. And we've also now have a video with a lot of persistence and patience on Rita's part, and Theo, who is the person putting it together. So that is done now. And as I said, we still have these permanent systems that we're looking at. So just quickly to explain the bioassay method, it's looking at the germination and growth of broccoli seedlings. Specifically, what you measure is the hypocaudal length. And on the bottom of that screen, you can see in one of the tests, the hypocaudals are cut and basically measured. But why are we doing this? So first of all, it's a very sensitive assay. Not all of the PGRs that we're looking at are actually measured in the lab pesticide scan. So we need some way of measuring what's happening there. And we're also looking for any other treatment side effects that might occur that aren't measured in any of the lab stuff. So that's why and how we're doing that. So just to an example of the sensitivity, this slide shows the standard curve of the paclobutrazole or the bonsai or piccolo bioassay. And you can see that even at 3 parts per billion, there is significant reduction of growth in those hypocaudals. So that's why we're using this. It's very sensitive. So this picture shows the pilot plants moved to the site for the COHA-ACHO project. The water that we're using comes straight from the greenhouse recirculation tank, so it's real greenhouse water. It's pumped into those white tanks at the back, and then through the series of these cells. So the current media treatment sequence, we always start with the hardwood chips in cell 1. And then there's a series of mineral media after in the other cells after that. So pea gravel slag mix. There's wollastonite. We're looking at filter sand. And we have added to this particular project the granular activated carbon, pardon me, because we know that it is going to remove the PGRs and the pesticides. It's very good at that. And so that essentially is our standard that we're measuring everything else against. So in 2020, we did the batch trials where we wanted to test each media on its own, how it performed. So the influent, so we spiked the influent, the greenhouse water with 4 PGRs. We used Bonsai, Medallion, Cyclocell, and B9. Then that spiked and mixed the heck out of it. And then that spiked water went into each of the cells. And we periodically sampled from those cells to get an idea of the effective hydraulic retention time. And we did a run in August and in late October to get a temperature effect, submitted samples to the lab for chemical analysis. And this scan measures, I think it's 483 particular compounds. So it looked for anything and everything in there. And then we did the bioassays. So an example of the lab results, this is in the first system, this is for paclobutrazol. And this is one of the ones that we spiked in, so the levels are high. So on the left, you see the dark green bar is what the concentration in the spiked feed water, and then what was removed. So the wood chips did a great job of early on removing the paclobutrazol, and so that was consistent throughout. The activated carbon did the same thing. Pea gravel really didn't do a whole lot, but wollastonite was really quite good at removing some of it, not all of it, but some of it. In the second system, again, the green arrow or the green bar on the left is the influent. And the wood chip is doing a good job. Pea gravel and slag not doing much at all. But the filter sand in this case is actually taking out quite a bit. And so that was the general pattern of the chemistry for most things that were in that water. There was a few exceptions. So in this case, there was a bit of flunicamide, which is an insecticide in the water. And so in this case, wood chips took out a fair bit, but certainly not down to zero. So it's, removing roughly 60%. And the activated carbon, of course, it's taking everything out. And what was interesting in this is that when you look at the second system, the thing that performed the best was actually the pea gravel and slag. And so it didn't take out everything, but it certainly reduced that level quite a bit. Oops, sorry. Okay, so what did the bioassay results look like? So this is one of the results from one of the runs, the October run in this case. The control, the nutrient control is on the left, so that's just receiving clean nutrient water. The reduction from the spiked influent is quite a lot. It's around 60% here. That's that red line that I just put up. And the wood chips and the activated carbon basically restored growth to the level. There's a fair bit of variability, but we had good growth in those treatment cells. The pea gravel and the pea gravel slag didn't do a whole lot, but you see here that one too many, that the wollastonite and filter sand did restore some growth, not all, but some. And so just quickly in summary, for the batch results, I've tried to reduce the complexity of this slide by just saying, okay, if it removed over 90% of something, then it gets a check mark. So that clears up the slide quite a bit. The top pale green ones here are PGRs. The pale gray are fungicides, and the pale yellow here are insecticides. And so you can see that the granular activated carbon takes out everything. That's what it's supposed to do. Wood chips were almost as good, with the few exceptions of, in this case, the flunicamide. Wollastonate was also quite good. Filter sand, not bad. Pea gravel and was not that great, but you wouldn't expect it to be. And then the pea gravel slag had this one instance of taking out something that the others did not. Well, the activated carbon did, but so it was the odd man there. So to get to the 2021 season this past summer, so we ran the series, did the series runs to test the media sequences, and again looked at operational parameters, and we did both the lab analysis and the bioassays, quite a bit of KTT, and as I said before, we looked a little at the permanent systems for nutrient fungal populations, because we want long-term data on these systems, and this is about the only way to do it. And we also squished out enough money out of this the analytical budget to be able to do a couple of samplings of the PGR and pesticides at those sites too. So the series runs in the pilot plants, the treatment media, it looks basically like it did for the batch studies. The only thing that was different was that we took out one of the pea gravel slag tanks because we didn't want to double treat. So that's really the only difference in how those were what we used, and the setup looked like this. So we had the two pilot systems, and each one of them, the influent water was spiked with a 4 PGR mix, the same one as we used for the batch. And then the water was pumped in from the feed water tank into cell 1, and then through each of the cells in sequence. And we sampled between each of these cells to get our samples for analysis. And again, we did an August and an October, November run to get that temperature effect. So what did the results from this look like? So I'll start with the bioassay. This is the August one. And so controls on the left, again, big reduction from the influent in length, the hypocaudal length, and the wood chips and the activated carbon pretty much restored growth. And then what is interesting here now, you have to think about this is now a sequence, like a series. And so you would expect that if the wood chips remove pretty much everything, then all the tanks following should give you the same sort of result. And here we're getting some inhibition, particularly in the wollastonate and the filter sand. So the question is, why the heck did that happen? Because you wouldn't expect that. Like once it's not going to grow in there. So once it's out, why is it seemingly coming back? So to answer that, we look at the chemistry now. And so this is the paclobutrazol, or the bonsai. So the first thing is that, yep, the wood chips are doing a pretty good job. They're in fact removing 98% of what went in there. So that's great. But what we see here, particularly in the wollastonate and the filter sand, you can see that there's still some paclobutrazol in there. And so what we figure is what happened, because in the batch study, each of those media was treated with the full strength influent water. And so while if you think back to what those batch slides looked like, you could see that the wollastonate and the filter sand were removing a fair bit of that paclobutrazol. So what we think is that what is happening is that the media, it's mineral media, and what it is doing is loosely binding these chemistries. And so that even though these systems are rinsed several times between runs, that there is essentially a leaching of the chemical from those mineral cells, as opposed to in the wood chips, it's probably breaking it down because that's a biological system, and that's what should be happening there. So I guess the moral of the story is start clean. but it does show that there can be residuals and loose binding of some of these materials. So in this massive summary slide again, the PGRs are the pale green. Metallochlor is a herbicide that's in this pinkish color. Gray is fungicides, yellow is insecticides. And again, the active after the GAC tank, there's nothing much left, and that's what that's supposed to do. Wood chips are really very good for the first, for the PGRs, the herbicide and the fungicide, a little less so for the insecticides. You can see that they are a bit of a harder nut to crack. And some of these other residuals where you see an increase is probably because of that leaching effect. So the permanent installs, what did we do with those? So I just want to show you some of the PGR or the pesticide results from that. Just a quick snapshot. So these are what the systems look like. You can see in this last one that there's some production on top of the system. And so we tried very hard at first, because when you take a sampling, because there's a residence time within that system, when you're sampling the input and the output on the same day, it's really not the same water that you're testing. So what we wanted to do was to try and find out exactly how fast does that water get from the greenhouse into the system and through the system. And so we spent a lot of time trying to do some rhodamine dye testing to figure that out, but it proved to be really complicated and not particularly successful. But eventually you could probably do it, but we just didn't have the time essentially to do that. So what we opted for was some snapshots. So throughout the growing season, what we did was to sample these systems, and I collected up all the samples and stuck them in my freezer at home. And then when at the November, because you have to submit a whole bunch of samples to the lab at one time in order to get the reduced price of just under $200 a sample. So we decided or selected which ones we thought would actually generate the best information in terms of PGRs or pesticides applied. And so we have a greenhouse in July and November, container nursery in July, and another greenhouse in November sampling. And so I struggled with how to present this data, but essentially I've reduced it down to just what was the number of PGRs or pesticides that were detected in the first or second cell, and then asked the question of whether or not they were reduced across the system. So what was it like in the last cell compared to the first couple of cells? And generally speaking, with a few exceptions, there was a reduction going across the system. And I know it's really non-data data, but the numbers and the types and everything are just so scattered that it's complicated to look at. So I thought this would simplify it, but essentially the systems are reducing what is going in there. So if they're designed and managed properly, do they remove nutrients? Just keeping an eye on my time here. So yes, but we have it, but they don't remove salts like sodium and chloride. I wish they did, but that doesn't seem to be an option. Do they remove pathogens? We've specifically looked at fungal populations, and so yes, they come out in the wood chip cell, but we haven't tested for viral or bacterial pathogens, so there could be lots more work done there. And then for this project, do they remove PGRs and pesticides? And the answer really is mostly. They're really very good for PGRs and herbicides and fungicides, but insecticides seem to be a bit more challenging, and that's a whole other research project that could be done there. And the caveat to all of this is that their performance is impacted by temperature and by flow rate. And there's other design factors such in permanent systems, such as cell depth, that kind of thing. And so there's still kind of lots more work to be done here, but really they're really very good for doing what we've asked them to do. And not last but not least is the video that was produced, and I don't know if Rita's going to talk about those or has talked about those, but probably more of you will be contacted, but there is a video now. I'm not sure when it goes up, but that's a question for Rita. So that was done too. So thank you very much. Acknowledgement to we have a large technical advisory committee, and we still have a meeting to do yet this year. And for additional financial support from Flowers Canada and Landscape Ontario and ODVG and Aqua Treatment and all of the growers, which is fantastic. And of course to COHA and Ag Canada. So thank you very much.